In this video, we will be inoculating agar plates from a bacterial culture in liquid media. We will first go over streak plating, then we will describe spread plating. We will be working with Escherichia coli strain MG1655, a common laboratory strain of bacteria. Note that ideal growth conditions, media requirements, and appearance of cells may differ depending on the organism used. Streak plating is done with the goal of obtaining isolated colonies of a single organism. As cells from a sample are streaked out on a plate, cells are deposited along the streak lines. Upon subsequent streaks, fewer and fewer cells become deposited, allowing deposition of single cells that will grow into isolated colonies. This allows for the study of a single, isolated species in pure culture. Subsequent applications of streak plating include sequencing, phenotypic characterization, and bacterial cloning. There are many different streak patterns used for this purpose. We will be demonstrating a zigzag pattern. Spread plating is most frequently done to determine the concentration of cells in a sample. As explained in detail in another JEMI methods video, dilutions of bacterial cells are done to obtain countable numbers of colonies on a plate. Samples are spread evenly on a plate to allow growth and counting of these isolated colonies. The number of colonies on a countable plate ranges from 30 to 300. Importantly, dilutions of cells must be performed in sterile saline or growth media to maintain an isotonic environment. Performing dilutions in distilled water may cause cells to lyse due to osmotic pressure differences between the cell and the surrounding liquid. Before plating your culture, it is advisable to pre-warm your plates at the desired incubation temperature. This will help ensure that the surface of the agar is dry, allowing the growth of discrete colonies. This will also prevent moisture and condensation from causing growth of undesired organisms. First, prepare your bench. Wipe down your bench with 70% ethanol or a similar disinfectant. Set up a Bunsen burner in the middle of your working area to provide an aseptic working environment 6 to 8 inches around the Bunsen burner, as described in another JEMI methods video. Place the samples, plates, and equipment required for your experiment around the Bunsen burner in an ergonomic fashion. Light your Bunsen burner by turning on the gas and applying a spark or flame. Flame your wire loop in order to sterilize it. The wire loop should be held at the tip of the flame until red hot. Remove the wire loop from the flame and let cool for 10 seconds. To make sure the loop is cool enough to use, touch it to the lid of a petri dish. If condensation appears on the lid, the loop is still too hot. Using the sterilized wire loop, carefully touch the bacterial growth you wish to plate. This could be a single colony from another streak plate, a liquid broth culture, or an environmental sample. Within the aseptic working environment, gently streak a small area along the edge of your plate. Sterilize your wire loop in the flame as before and allow it to cool. Drag your wire loop gently through the previously streaked region and streak approximately 1/3 of the plate in a zigzag pattern. Sterilize the wire loop again, let cool, and drag your wire loop through the end of your second streak, continuing to streak another third of the plate in a zigzag pattern. Repeat this process at least one more time until no unstreaked space remains on the plate. This will usually take 3 to 4 streaks to accomplish. Be careful to avoid touching the original streak, which has the highest number of cells. Replace the lid and place the plate in an incubator in an inverted position at the desired growth temperature and conditions for the desired length of time. In our case, we are streaking out laboratory-adapted Escherichia coli and will be growing it overnight at 37 degrees Celsius. Remove the plate and observe the growth. A properly streaked plate should have isolated colonies on one of the zigzag streaked patterns. Light your Bunsen burner by turning on the gas and applying a spark or flame. Using sterile saline or growth media, prepare dilutions of your desired liquid culture. Note that turbid cultures will usually result in excessive bacterial growth when spread plating, and that multiple serial dilutions of culture may be required to obtain colonies that can be accurately isolated and counted. Here, we are performing 1 in 10 dilutions of the culture by adding 100 microliters of culture to 900 microliters of sterile saline and repeating several times, ensuring samples are thoroughly mixed each time. Prior to plating, vortex your sample to ensure it is well mixed. Pipette 100 microliters aseptically to the middle of a plate. Dip your glass spreader into the 70% ethanol and pass the spreader through the tip of the flame until the spreader is dry. Allow the spreader to cool for 10 seconds. Using the spreader, push the sample around the entirety of the agar plate, rotating the plate in order to obtain entirely uniform growth. Replace the lid and let the sample absorb to the surface of the plate. This will take 5 to 10 minutes depending on the volume of sample used. Place the plate in an incubator in an inverted position at the desired growth temperature and conditions for the desired length of time. Remove the plates and observe growth. Our undiluted sample shows confluent bacterial growth, while our diluted sample shows uniform growth of isolated colonies.